The human body is a complex wonder of evolutionary progress. It is capable of feats and strengths, exploration, and can protect itself from illness. The body as a whole is an emergent property of many smaller parts, like our organs, and the cells and proteins that make up the tissue that make up our organs. The most outer layer of the cell is defined by its phospholipid bilayer that is semi-permeable, keeping unwanted items out of the cell and protecting the most precious content of the cell, our genes. However, as protected as we might seem from unwanted items, the environment around us can have monumental effects on our body's health. Everything from the water we drink to the chemicals we work with and the air we breathe can expose us to many different substances that can pass through the body's various defense systems and alter our genetic makeup and expression. The field of study concerned with understanding these interactions is known as toxicogenomics. Traditional methods in toxicogenetics focus on determining toxic potential and evaluating the risk of chemical substances. This is done mainly through the examination of the different biochemical pathways connected to responses observed have on gene expression, proteins, and metabolite expression, which results in mutations in cells, tissues, and organs. Methods like the real-time quantitative polymerase chain reaction and microarray analysis are used to determine transcription changes in DNA after a cell interacts with a toxic compound, therefore being able to predict the adverse effects of a compound in biological systems. So, if the lesson we learn from toxicogenomics is that carcinogens and prolonged chemical exposure are bad, then shouldn't we just avoid toxic chemicals at all costs? Why do we need a field of study dedicated to something so obvious? Well, it's not that simple. Many of the products we use throughout the day contain chemicals that we think are safe, but turns out that they can be pretty harmful. Also, waterway contamination and exposure to certain metals or chemical byproducts in construction may not immediately result in disease until years later. For example, journalist Eric Ortiz reported on the unusually high number of cancer cases in Mooresville, North Carolina. By tracking the cases of thyroid cancer in her neighborhood, Susan Wynn was able to determine that something unusual was happening. She suspected the nearby lake had something to do with her daughter's and neighbor's cancer. With the help of Duke University, they are now investigating why their area is a cancer cluster. As Ortiz reports, the suspected link is coal ash leaked by Duke Energy's online storage basins. Ortiz does point out that the North Carolina State Department of Health and Human Services states that, quote, there are no published studies to support an association between coal ash exposure and thyroid cancer. But a study by the New York University School of Medicine in 2016 found that chromium, a common component of coal ash, resulted in the loss of acetylation at histone H4K16 after exposure. Ultimately, what they found was that chromium oxidizes iron, which activates enzymes capable of modifying DNA, resulting in the overexpression of the NUPR1 protein. The overexpression of this protein has been repeatedly shown to be involved in transforming healthy cells into cancer cells. There are many more cases like this that require dedicated scientists to focus on the investigations in order to answer the questions presented by the field of toxicogenomics. So, as you explore possible careers in genetics or move on to another scientific field, keep track of toxicogenomics and all of the new information it has to offer.